All right, today we're diving into something called Amplicon sequencing. It's this super powerful way scientists can zoom in, like with incredible precision on very specific parts of the genome. Let's get right into it. So, let me ask you something. How would you find a single tiny typo in a book that has 3 billion letters? I mean, that's the scale we're talking about with the human genome, and it's a real challenge researchers face every single day. So how on earth do they pull it off?
Well, the answer is actually pretty clever. Instead of trying to read that entire, massive library of a genome, Amplicon sequencing is like having this, well, a magical photocopier. It finds the exact page, the specific gene you're looking for, and just makes millions and millions of copies of it. And when you do that, that one little typo, or what we call a genetic variant, suddenly becomes super easy to spot.
And that copied page? It actually has a name. We call it an amplicon. Basically, it's just a piece of DNA or RNA that's been artificially copied over and over in a lab. It's purpose-built for analysis. And really, these amplicons are the secret sauce that lets us focus our genetic searchlight with such insane precision.
Okay, so this genetic photocopier idea sounds cool, right? But how does it actually work? Well, it all boils down to a really revolutionary lab technique. You've probably heard of it. It's called the polymerase chain reaction, or just PCR, especially over the last few years, right? Well, PCR is basically the engine that makes all those amplicons. It uses this simple repeating cycle to churn out millions of copies from just one single starting piece of DNA. The cycle is just 3 steps. It's pretty straightforward. First up, denaturation. You heat things up, and that unzips the DNA double helix into 2 single strands. Then, step 2 is annealing. Here, you cool it down a bit, and these little DNA pieces called primers, think of them like bookmarks. They find and lock onto the exact spot you want to copy. And finally, extension. A special enzyme comes in and builds a brand new DNA strand, starting right from that primer. And you just repeat this cycle, 30, 40 times. Each time it doubles the number of copies, and you end up with this exponential explosion of your target sequence. It's pretty amazing.
Okay, so making millions of copies is one thing, but why is this so much better than just trying to read the whole book at once? Well, it all comes down to the power of focus.
To really get it, let's compare it to something called whole genome sequencing, or WGS. Now, WGS tries to read the entire genome, but it does so at a pretty shallow depth, maybe reading each spot about 30 times. Amplicon sequencing, on the other hand, just completely ignores most of the genome to go ultra deep on a few specific targets. And what that means for you is less data to sift through, lower costs, and way, way higher sensitivity for finding those really rare, hard-to-spot variants.
And when I say ultra deep, I'm not kidding. We're talking about reading a specific region over 1,000 times. That kind of depth gives researchers the confidence to spot the absolute rarest genetic mutations, or maybe identify one single type of bacteria in a really complex sample. It's kind of like the difference between just skimming a chapter and studying every single word on the page.
And because it's so focused and powerful, this technique has become a total workhorse in tons of different fields. Scientists use it to profile the bacteria in our gut, to pinpoint tiny genetic changes called SNPs, to double-check that their CRISPR gene edits worked, heck, even to diagnose diseases by spotting a pathogen's DNA. The applications are just huge. So yeah, this is an incredibly powerful and precise tool. But getting it right, from designing the experiment all the way to analyzing the data, that takes some serious expertise and the right tech. And that's really where having a dedicated sequencing partner can make all the difference in the world.
Think about it. From designing the perfect primers for your PCR reaction to making sense of the massive flood of sequencing data that comes out, every single step has to be perfect if you want results you can trust. It's a really complex process, and having specialized experience is a huge, huge advantage.
And that's where a service provider like CD Genomics comes in. They offer that critical end-to-end support. They can basically guide a project from just an idea in your head all the way to the final data report, giving you that technical backup you need at every single stage.
And a huge part of that expertise is just picking the right tool for the job. Services like CD Genomics give you that flexibility with different sequencing platforms. So let's say you're looking at shorter DNA fragments, maybe for SNP detection. The Illumina platform is perfect for that, super high accuracy. But what if you're looking at longer targets, like a full-length microbial gene? Well, then the PacBio platform is what you need to get those long reads and see the whole picture.
The whole process is usually really streamlined. It pretty much always starts with a project consultation, just to make sure everyone's on the same page about the goals.
Then, after your samples pass a quality check, the lab gets to work preparing the sequencing libraries. After that, your samples get sequenced, and finally, a team of bioinformaticians crunches all the numbers to deliver a super comprehensive report.
But wait, what happens after all the sequencing is done? I mean, how does this mountain of raw data actually turn into a real scientific discovery? This, right here, is the magic of bioinformatics. It's all about taking those millions of raw genetic reads that come off the machine and transforming them into clear, easy-to-understand, and actionable insights that can actually help researchers answer their big biological questions.
So a final report. It's not just some single file, it's a whole package. Of course, you get the raw data, but you also get all the quality control metrics, a detailed report on any variants they found, notes on what the genes might actually do, and a whole suite of awesome data visualizations so you can really explore the results for yourself.
And honestly, these visualizations are what really bring the data to life. Instead of just staring at tables of numbers, you can actually see things like heat maps showing which microbes are most abundant, or these cool cladograms that reveal evolutionary relationships. It's all about making that invisible world of genomics totally visible. So there you have it. From finding that one single typo in a 3 billion letter book to mapping out entire microbial ecosystems, Amplicon sequencing gives us this incredibly focused and powerful window into the genome. And that really just leaves one question, doesn't it? What amazing discovery is hiding in your samples?